Thank you. This is a talk about microRNAs and microRNA binding. How for a very long time we've been looking at our benchmarking data sets completely wrong and misleading the whole field, my group, for a few years. So this is the story about how we figured it out and tried to correct it. Everything I'm showing here is mostly the work of these people who work really hard on it, and it's a collaboration between the University of Malta and Central European Institute of the Czech Republic. So if you need anything, if you have any questions, please feel free to send me an email, and I will be very happy to talk about this. So the story is a story about microRNAs. It's a story about neural networks, and it's a story about benchmarking. And I was very happy last year when basically the whole field of the Nobel Prizes was somehow relevant to this story. I felt like also here I see a lot of benchmarking work shown, and the keynote today was really hit very close to what I'm going to talk about, especially the idea of using these stupid models to try and see what is the baseline of your work. Okay, so what are microRNAs? Just a quick recap for anyone that is not an RNA or small RNA person. Essentially, these are small, very 20, 25 nucleotides long that get loaded into this protein of the Argonaute family, and then the protein uses the small RNA to try and find targets in other RNAs, mRNAs mostly, to try and figure out binding sites with non-full rules. So we don't exactly know the rules. There's some ideas about what some of the rules of binding might be. The seed matching at the 5' end of the microRNA, meaning consecutive stretches of approximately 6 or 7 nucleotides that match perfectly, and this kind of nucleates a binding. But we know that these seed binding sites only explain about half of the interactions that we see experimentally. So there's a big, let's say, field about actually figuring out other rules. So the task, very simplified, is if I give you the sequence of a microRNA and the sequence of a putative target site, can you predict if these two things bind or not, or how well do they bind? Okay, so the data sets that we use for that come from, I will not go too deep into the biology and the experimental part, but the data sets basically come from these things called chimeric reads, which are essentially sequencing reads that manage with a trick to capture together the microRNA and the end part of the target. So we can sequence in one read both of the parts of the interaction and then map them on the genome, etc. So we get these reads that have both, and then we can basically try to predict, try to build a machine learning model to predict this interaction. The problem is the experiments only give us positives. We don't have negatives. So we need to make our own negatives for this classification task. So basically the first thing that we did in this study was to analyze some new data sets that came out and produce a very big data set with about 1.5 million positive samples, positive interactions, while the previous biggest data set was about 5,000. So this is a orders of magnitude increase. But the problem of negatives still exists, right? So this is how our data set looks. The microRNA sequence about 20 nucleotides, target sequence 50 nucleotides, as binding, 0 is non-binding, 1 is binding. So how do we pick negatives? What we were doing before, and pretty much I think most people in the field have been doing, was this: like, we take a positive, it has a microRNA and the target site. we keep the target site, and we pick a different microRNA from the pool of microRNAs that were in the experiment. And then we match these two things. It's not guaranteed that they don't bind, but by and large, it's not an interaction we had seen before. And then we call that a negative. And this creates a huge problem, which we didn't know up to now. So we tried to make this bias model, like these stupid models that shouldn't be able to predict anything, because what you need to predict is the interaction of the two molecules. So if you make a model where you give it only one of the two parts of the interaction, only half the information, it should predict a 0.5 random, right? What we actually saw is that the microRNA only model actually predicts with an average precision score of 0.75. This approximately of it as a precision recall area under precision recall curve. So this means that just by looking at what microRNA sequences are in the positive and the negative set, it could tell, it could basically classify. So why is this happening? Is because, as you might have guessed now, is because there are some microRNAs that are very prevalent in the positive set. Let's say, imagine there's a microRNA that has 20% of the positive set. And then in the negative set, it will only be picked randomly, 1 in n times, right? N being the number of microRNAs that are in the hundreds. So in the positive set, it's 20%. The negative set is maybe 1%. And then obviously the model learns, if I see this microRNA, it's probably a positive. That wouldn't be a huge problem if we didn't see that this can affect actually the target prediction programs that have been published, we and others have published. And we see here that if we take an older test set and we, again, this average precision score, this is how different machine learning models do. And, oh yeah, sorry. And also, this is not only a problem of our data set, of this data set, for example. We see that in completely unrelated data sets that other people have been using as well. This is a completely different data set, different experiment, different type completely. So yeah, so we decided to correct this by basically flipping the shuffling. So instead of keeping the target and picking a different microRNA, we keep the same microRNA, so we're guaranteed to have the same frequency there, but we now pick a random target. Why this is not a problem, as you would imagine now we're flipping the problem, is that the targets are actually very variable. You don't get the same target twice, essentially. Very rarely you get the same target twice in our data set. MicroRNAs, on the other hand, there's only a few hundred of sequences, and they over 1.5 million, they're found multiple times. And then when we correct the data set and we rerun all these trained models, we see drops in the average precision score up to 10 points. And somehow this, sorry, took over that. But what I wanted to point out here that you cannot see is that these three methods there, the yellow ones, were actually published before this data set was created. So before this experiment was even done. So these have somehow learned this bias without, for sure, never have seen that data set because they were published before. So we have a big problem, right? Because how can they know the bias of a data set that will exist in the future? The problem, the big problem is that many of these data sets are actually produced from the same cell line, and this flipping T-Rex 293 cells. So this original paper was published in 2013, and this is on an AGO1 protein, which is in the same family, also use microRNAs, etc., for many years, there was no other data set. So many people have taken these targets and tried to build models. This is done in the same cell line, which means the frequency of the microRNA's expression is the same because, it's the same cell line. Then this has been filtered through different target databases, etc., and then it always finds its way back into different data sets that people build, and in general creates this issue. So this is one. Then the other ones that I showed before, the other data sets have also now been used, and we have created, let's say, this mess. Okay, so now to the good part where we correct it, right? So we have corrected this this issue. We have created new data sets. We have created also 4 testing data sets. Importantly, the last one called the Manakov left out is a part of this big 1.5 million data set from microRNAs that were not found in any of the other data sets. So these microRNAs were, because so deep, we could find microRNA targets from microRNAs that were not in any of the other data sets. So this, and we didn't put any of these microRNAs in the training set either. So these are, this, let's say, a left out data set. So now we benchmarked all these models on this. You can see all of these in the paper, but I'll show you now this test set, this Manakov test set, and we see that all the models drop a lot. Only the target-scan CNN model, the pink one, doesn't drop because this was never trained in this type of data. This was trained in a completely different type of data set. These dots, circle dot symbols, this is a naive model that would use just the number of, just if there is a seed or not. So remember, the seed is a consecutive stretch of fully binding nucleotides. So you see that even the best models don't maybe barely outperform like just counting seeds. So this means to me that we haven't really learned much in this task. Okay. So what we do is now we take one of these models, one that we have previously published, and we retrain it on the new data, on the new data set. And when we retrain it on the new data set, you can see the last line there. We jump essentially from 0.7 to 0.84, which means it can learn quite well. I mean, it improves. Just to give you an idea on the previous figure I showed, on the left there, this is the green line. It's this line right here. And this has been retrained with 2 of the new data sets. And these are the blue and the green line up there. So the blue line is the one retrained with the 1.5 million, the big data set. So we see, let's say, an improvement. The goal of this was to do the benchmarking, not so much to do the new model. Now we're working follow-up to actually optimize the model and do hyperparameter tuning and different types of representation, etc. Another interesting point is that we started basically with taking this big data set and using subsets of it and seeing if we just use 1,000 samples, how well can we learn, and then increasing the size. So this is a logarithmic scale here. And you will see that at about 10,000 samples, positive samples, we kind of hit a plateau. There's an obvious plateau there, and then the increase up to 1.5 million is much less. So it seems like at least that model, right? I'm talking about this one specific model. It seems like that model hit a ceiling, kind of. I mean, it's learning a little bit, but that doesn't mean that different architecture, someone smarter coming in with a different representation coming in could very well get more information out of that, right? So the data sets are now publicly available. We try to make it as easy as possible. we have actually re-implemented a lot of the encoders and the models that other people produce, all these other systems that I showed. So if you want to like benchmark your model, you can really do it really easily. You can also use the parts of ours and other people's methods. There's a Python package that you can use within Python, or you can just download the data sets, directly. And now a plug to our poster. So we also, instead of having what was the semi-intelligent PhD student try, we actually tried an agentic system that we've been developing on this data set as well. And so our best human state of the art was 0.86 here. The agentic system gets to almost 0.78, which is better than pretty much all the other tools we had made previously, which is pretty nice considering it doesn't know what any of this is. So if you're interested in this, there's a poster to then tomorrow downstairs. Come find us. And that's everything from me. Thank you again, and any questions?
So much. We do have some time for questions.
Very nice presentation, very nice work. Let me say, what is the distribution of different microRNAs in your thousands of targets?
Yeah, there is, I mean, I don't have a figure with the distribution, but I can tell you there's like one microRNA with about 20% of targets. Then, the next few have in the, let's say, 5%, the next like 10 or 20. And then there's a very long tail of few, below 1%, let's say, or below 0.1%. So it's a very kind of long-tailed. But if you take, I think the top, I don't remember, 10 microRNAs or something, you're probably explaining about 50% of the data, or 30, 40% of the data. So that's why there was such a big issue. And that's actually the same issue with many other data sets, because people tend to validate targets of the same microRNAs. Like if you see Let7 validation, like tons of them. But if you have an obscure microRNA, not many people will validate it. It will not be represented so much in the highly expressed and well-known disease-associated microRNAs get preference. Yeah.
One more question.
Hey, thank you very much, Panagiotis, for looking into data that many people have been using, right? And clearing this up for the field. I was wondering, this plateau that you were showing, where you said, this method doesn't get better if you add more data, whether this could also have to do with the diversity of microRNAs, that maybe, there's some rarer microRNAs that are not similar to other family members, or I don't know, something like that, maybe?
Right. So that's why I put down their microRNA-specific rules. I didn't really go deep into that, but there is some evidence. I mean, there's some talk, let's say, I don't know if there's real evidence, that maybe different microRNAs have slightly different rules. And to learn that, maybe that's what we're missing in a way, right? To learn that, you would need maybe microRNA-specific models, or a much larger representation of maybe because of the mixture. This is learning the highly expressed microRNAs much more, and then maybe these rules don't generalize within the same, like with different families. We really don't know that, and it's an open question. So,
Quick question, Laura.
Yeah, just to comment on that a little bit, could you potentially leverage the secondary structure of the microRNAs into this? So like if you predict the secondary structure, would that maybe improve the performance?
Yes, this is a great question, actually. And if anyone is a structural person, talk to me, because I'm interested in that. There have been some attempts in the beginning, like many years ago, of doing co-folding. The problem is that this is all done inside the protein. So the co-folding needs to happen with the structure needs to be predicted under the constraints that the protein gives, right? It's all in a pocket of a protein. So if you put, so I tried putting some of these samples in AlphaFold, where now you can put a protein and 2 RNA sequences. But the problem there is that because AlphaFold is kind of trained on things that actually bind, it will really try to force things to bind together anyway, right? So it's, I'm not so much a structural person, but it seems like there's definitely information there, but we don't know how to model it. So yeah, it's a very good point.
Thanks so much, Panagiotis.
Thank you.